Today we're talking about Chapter 3.3 and Chapter 3.5, which is going to focus on the structure of the cell membrane and how it is used to transport materials. You might want to take a look at the Amoeba Sisters video that's linked in your notes presentation online. I'm not going to include it as a part of this video because you can watch that separately, but it does cover much of the same information, and it is a really good look at the structure of that cell membrane. So again, check your notes that are posted online and you can get the link to that video. Now what you are starting out with is a picture of the cell membrane that looks like this, and it has several different parts. And what you are asked to do is to color code the specific parts of the cell membrane. So what I would suggest doing is go ahead and just watch this segment of the video and kind of take notes on what part is located where, and then you can pause the video and go back and you can add your colors. So what we're looking at here, first of all, is a double layer of phospholipids. We have the polar heads, which are all of the circle parts here for your phospholipids. And then the wiggly parts at the bottom, those are fatty acid tails. So that would be the non-polar region of the cell membrane. You can see that all throughout the interior is going to be non-polar because all of those squiggles are fatty acids. Embedded in that membrane are some of these little molecules here. They have a bunch of like hexagons and pentagons all attached. Those are molecules of cholesterol. They are found on the interior portion of the cell membrane, and they are a type of lipid. So they're hydrophobic and they stay associated with those tails of the phospholipids. They actually play a really important role. They help maintain the fluidity of the membrane. It prevents the fatty acids from packing too tightly together, but it also helps to attract them to each other so they don't get too far apart. So it stays just right at the right fluidity for the cell to be able to function. Also embedded in this membrane, we can see there's a large protein here. We'll start with this guy. This is a transport protein, and I can tell it's a transport protein because particles are moving through it. All of these little triangles represent some kind of particle that's outside of the cell, and they are diffusing through this transport protein down their concentration gradient to the inside of the cell. There's some other proteins that are associated here as well. This represents a protein on the inner part of the membrane only. We can call that a peripheral protein, which peripheral just means off to the side. And this is probably something like an enzyme. An enzyme that is typically going to be acting inside of the cell might be associated with the cell membrane, but not necessarily going through it like this transport protein does. And then finally, the other protein that we have is this blob up here. That is a part of a protein that is embedded in the cell membrane. And notice that it has this carbohydrate chain sticking off. So we would call this a glycoprotein, and glyco refers to the carbohydrate. Protein refers to this bottom part. So this would be the protein portion. This would be the carbohydrate portion. And the purpose of that is to serve as a marker to help identify a cell type. So these are kind of like flags, like you have a flag for different countries. Well, cells have flags that are made out of these carbohydrates, and the shape of the branches of that carbohydrate helps identify a cell type. Sometimes you can have those carbohydrates attached to a lipid. So this would just be a single lipid here. If it has carbohydrate attached to it, we would call that a glycolipid because, again, glyco refers to the carbohydrate, and if it's attached to a lipid, we would call it glycolipid instead of a glycoprotein. So those are all the major parts of the membrane. So go ahead and go back and rewind the video if you need to. Go back and replay it to see and make sure that you have everything properly color-coded in your diagram. And now we're going to talk about how that membrane actually functions. It is a very fluid structure, meaning it's flexible, and that means that it can actually form these pockets of membrane that we would call vesicles. If you remember that from your previous studies of the cell, you know that a vesicle is a membrane-bound bubble, essentially, that is inside of a cell. And you can see how that forms if you look at this diagram here. This portion of the membrane is budding upwards. So if we're trying to package something inside of it, if that membrane bubbles up and then we connect the bottom to form a complete circle, then that circle right there would be the vesicle, and the membrane, the rest of the membrane will reattach to itself. So this would be like the cell membrane, and this would be the vesicle that it just created. You could also go the other way. You can have something coming into the cell. So maybe this is a vesicle that's being received by this cell. So it would approach the membrane, it would fuse with the membrane, and it would join together like what you see here. And then that membrane would flatten out, and whatever was inside of the vesicle would get dumped into the cell. So those two processes of moving things in and out of a cell using a vesicle like this are called endocytosis and exocytosis. Endo, you know already, that means inside. Exo means outside. So if you are moving something into a cell and you are fusing that vesicle with a cell membrane, we call that endocytosis. And for exocytosis, it would be like if we were looking at this diagram and going to the left. So a cell is producing something and it wants to send it out of the cell. It's going to pinch off a part of its membrane and package that material inside. And then this would be the vesicle that contains that product that needs to be shipped out of the cell and taken somewhere else. Here's a good comparison of those two processes side by side. So again, we have the cell membrane here. You'll notice it's called a plasma membrane. Some textbooks call it a plasma membrane. Some call it just a general cell membrane, but those two things mean the same thing. There's no difference between a plasma membrane and a cell membrane. So on the left side, exo, meaning it is leaving whatever product is being produced down here in the cell. You can see the dots are the product that are being produced. Inside of the cell, there are organelles that will package up that product into a piece of membrane. That membrane will pinch off and form an individual little vesicle right here, and the vesicle will fuse with the cell membrane and eventually open up and release those particles. You can see those particles are leaving the cell. On the right side, we have endocytosis. This is really common in single-celled organisms. When they eat food, they actually just use their cell membrane. If it's a single cell, it doesn't have an entire digestive system, but it does have a membrane. And so this food particle can be surrounded by membrane. It pinches off into a vesicle, and now that vesicle can go into the cell, and eventually the contents can be digested and used as a food source. Now, there are three types of endocytosis based on the particles that are coming into the cell. The first one that we have here is pronounced phagocytosis, and that is what we just talked about on the last slide. If we have a single-celled organism that is essentially eating a food particle, that solid particle being taken into the cell, whenever a cell brings in a solid particle like that, we call that phagocytosis. The phago part refers to that it's eating a food particle. The second one that we're looking at is called pinocytosis, and if the first one, phagocytosis, was cell eating, pinocytosis would be cell drinking. This would just be if there is fluid outside of the cell, and there might be some solutes that are dissolved in that liquid, but the cell is just taking in a pocket of fluid. So instead of being a solid particle like we have in phagocytosis, it's now bringing in liquid in pinocytosis. And then the third one is called receptor-mediated endocytosis. And this one is how a cell is able to be picky about what it brings in. There are certain receptors on the cell membrane. You can see them here with those green shapes. And the receptors can pick up certain types of particles from outside the cell. And when they bind to those particles, it's going to hold the particle right here in this area next to the membrane, but it's not going to attach to these blue squares. Those blue squares are not needed by the cell. So only these stars are going to attach to the receptors. So when this membrane bubbles in, it pinches off and forms a vesicle, we have the stars collected inside of that vesicle, and we don't have those squares. And so again, the cell can be picky about what it's bringing in if it has receptors that are specific to the type of particle it needs to bring in. Those vesicles that we just talked about need to be able to move around inside of the cell. So I wanted to show you this little animation here. This is a computer animation of an actual the way that a vesicle would look, the way that it would be moved around inside of a cell. So every cell has inside of it kind of a skeleton. We call it a cytoskeleton. It's a network of fibers, and it's basically like little train tracks throughout the cell. And a vesicle, when it's moving, it's going to travel along that track. This protein that you see here that looks like it's walking and actually taking steps, every time it binds and detaches, like taking a step here, that is using ATP. So this protein, its job is to carry that vesicle along this track of the cytoskeleton, and that's how things move around in the cell. It does require the use of ATP. So anytime you have a vesicle, whether it's moving into the cell or moving out of the cell, that is something that is going to require the input of energy. So in terms of the types of transport, we'll talk about active versus passive transport. Active transport is the type of transport that requires energy. So both endo and exocytosis would be an example of active transport. The last thing that I will leave you with here, there's just a short video clip. I'll go ahead and play that. There's no sound associated with this video. It's just a video through a microscope. And what you're looking at here is an amoeba. And an amoeba is a single-celled organism. It's famous for just living in pond water. And it's also famous for being this kind of a blob-like shape. So what you see here were those pseudopodia, that false foot, pseudo meaning false, pod meaning foot. They surrounded a little tiny cell right here. You can see it wiggling around right now. But the amoeba completely surrounded that cell because that cell is going to be food for the amoeba. So that would be an example of phagocytosis happening because the amoeba is surrounding a solid food particle with that membrane, and now that solid food particle is contained within membrane inside of this amoeba cell, and it will eventually be digested.